Now I would like to invite Professor Mark Hanson. Professor Mark Hanson, he is going to give us a lecture about the developmental origin of health and disease. Professor Mark Hanson is the founding director of the Institute of Developmental Science at the University of Southampton, director of the academic unit of Human Development and Health in the University Faculty of Medicine and British Heart Foundation, professor of cardiovascular science. Welcome, Professor.
Thank you very much indeed, and thanks for the invitation to sunny Dubai. It's certainly better than Southampton, where the temperature last night was 0 and where it's raining. So my task is made a lot easier for you this morning by the eloquent lecture that you've just heard from Professor Simmons, introducing what's now called DOHAD, the Developmental Origins of Health and Disease. There's now an international society for DOHAD, so I'd urge you to consider joining that. And I'll tell you a little bit about some of the upcoming activities of DOHAD at the end if I have time. This picture could be taken in almost any city in the world, couldn't it? And of course we worry about the effects on these two ladies of being overweight. But then we also begin to worry about the effect on the newborn baby, the child in that stroller that one of them is pushing along. And we worry about whether the whole story relates to the consumption of this sugar-rich drink that one of the ladies is carrying. I'm going to tell you that this is indeed an important consideration, but that it's not as simple as that. Many of the ideas that I want to share with you come from discussions I've had over the years with my colleague Peter Gluckman, who works in Auckland, and this was a book we published earlier on this year addressing some of the issues. So we might start by asking the question, what can we do about obesity? Where do you go when you want a question answered? Well, of course, you go to Professor Google, don't you? And this is a couple of weeks ago, what I found in response to the question, how can I lose weight? Not that I want to lose weight, but how can I lose weight? And you'll see an enormous number, millions and millions and millions of hits. They can't all be right, can they? Actually, when you look at them, hardly any of them are right. So what on earth is going on here? Why don't we know the answer? Well, I think until recently, we've been shooting at the wrong target. And a lot of discussion about obesity and non-communicable diseases, particularly type 2 diabetes and cardiovascular disease, but also some forms of cancer, has been dominated by the paradigm that it's all our fault as individuals, that it's our voluntary consumption of too much to eat, with too much fat and salt in it, not exercising enough, as well as exposing ourselves to toxins such as chemicals. Those of you who heard Professor Wald yesterday heard him refer to the absence of a strategy for public health in relation to folate fortification of flour, saying, well, it's up to individuals to take folic acid to prevent neural tube defects and so on. There's a similar nanny state fear going on here globally in relation to the obesity epidemic. But we have to recognize that this strategy is not working. It may work for smoking, but actually the rate of obesity and indeed morbid obesity and the prevalence of NCDs continues to increase, especially in this part of the world. And in fact, when you look at many of the research studies that have been done on animals, the kinds of things that Professor Simmons referred to, and indeed in humans, we're just beginning to get data that appetite, what we want to eat and how much, is set very early in our lives. And so it may be that these interventions are coming too late. And in any case, if we think of low and middle income countries, is it really appropriate for sub-Saharan Africa, where NCD risk is increasing enormously, to say they should eat less and exercise more? I don't think so. So is the problem really that lifestyles are difficult to change in adults? That might be the problem. Or is it that there are more fundamental drivers? And so when we were asked to comment on this for Science Translational Medicine, Our answer to the question, why are we losing the war against obesity, is that we intervene too late. And this very simple excuse me, diagram shows the strategy that we've developed over the years. There's nothing very complicated about this, but I think it comes as a surprise to some adult physicians. The risk of non-communicable diseases increases throughout the life course, but in a non-linear way. That's because our plasticity as humans declines from the developmental phase onwards, and so our responses to the new challenges that we face every day, from diet and so on, become increasingly inadequate. You can't bring someone who has a high-risk trajectory down to zero by an intervention or treatment in adult life. And I'll show you in a moment that even screening here in people who are very seriously at risk only has a mild effect on outcome. So don't we need to think earlier about interventions in childhood and adolescence, in the mother and infant, and use some of the biomarkers, the epigenetic changes and other things that Professor Simmons has shown you a minute or two ago, to identify those who are at risk and to think what we can do. You've seen this data already from Becky Simmons' talk, the data from David Barker's group, showing that birth weight is a risk factor for metabolic syndrome in adults, these adults born in Hertfordshire in the county of England around the time of the Second World War. The more I look at this slide, the thing that strikes me more and more is the fact that this is a graded effect. Yes, the low birth weight babies had the highest risk as adults, but it's graded across the entire range of normal birth weight and probably actually would increase again at the high birth weight range here in modern populations. So we're not just dealing with the small baby, the IUGR fetus, we're dealing with a graded risk, a graded steepness of that trajectory across the population. I mentioned a moment ago screening, and I just draw your attention to this very interesting study that was published in The Lancet just a couple of weeks ago, where they looked at a very large study, over 16,000 adults in the top 25 risk distribution for undiagnosed diabetes, did screening on them or not as an RCT, and then did a 10-year follow-up of the hazard ratio for all-cause mortality. Here's the data. You may not be able to see it at the back, but it doesn't matter. There's no difference between the screened and the unscreened individuals in terms of mortality, increasing cumulatively here on the y-axis over a 10-year period. Obviously, the screening has come too late to assist the outcome in these individuals. So let's then ask, in early life, which factors are particularly important? We're going to be hearing a lot after the coffee break about nutrition. And so I'll take my cue from that and say something about particularly maternal nutrition in pregnancy or even before pregnancy. In Southampton, we're doing a range of prospective studies, very difficult, very time-consuming, very expensive, but necessary if we're going to follow through the time course of disease risk in the way that I've shown you in that diagram. And one of the things that we did was to try to grade diet, not just to say, well, it's healthy or it's unhealthy, but to grade it across a score of what we call prudency, where the least prudent diet is the diet that, to be honest, we all love to eat. It's high in sugar and white bread and red meat and crisps and confectionery, and low in fruit and vegetables. And the most prudent diet, of course, is the opposite. So some years ago, we asked the question, what actually sets up the very early phases of that trajectory line I showed you in relation to mother's diet and the deposition of fat in the fetus? Guttorm Haugen, an obstetrician from Oslo in Norway, came to work with us, and he looked at the shunting of blood through the ductus venosus, bypassing the fetal liver, in relation to fat deposition in the baby. And here he's showing the shunting of blood through the ductus venosus, liver blood flow, and then relating it to mother's diet. And I hope you can see that those women who consumed the least prudent diet before pregnancy and in early pregnancy had fetuses in late gestation, 36 or so weeks later, where the blood flow to the ductus venosus was reduced. If less is going to the heart and the head and neck, where's it going? Well, it's going to the liver, and so increased blood flow to the liver was found. That matters because when we then went to look at adiposity in the children at birth using a DEXA scan technique, we can see that at any level of mother's adiposity, of course, more adipose women tend to have fatter fetuses, but at any level, those fetuses that had a higher liver blood flow, so a metabolic effect on the liver and a reduced ductus venosus shunting, were fatter. And now, most recently, we've seen that that story plays out even in those same children when they're 4 years of age. So mother's diet, pre-conception and in early pregnancy, is indeed setting up a risk trajectory, at least in terms of body fat of these babies. That's interesting because it harks back to some beautiful studies done by the diabetologist Ranjan Yajnik in Pune in India, where he came up with this idea that Indian babies, which we know are at high risk of diabetes, as Professor Simmons has shown you, had something funny about their body habitus at birth, and he called them the thin, fat babies. They were thin because they had relatively little skeletal muscle. There's one of the risk factors for insulin resistance, as you've just heard. But they were fat because they tended to have, compared to UK babies, more body fat, and the body fat was intra-abdominal, bad fat producing lots of nasty cytokines and giving them a high risk for disease later on. So under these conditions of undernutrition and increasingly overnutrition in India, we're seeing an increase in the numbers of thin, fat babies. And in fact, when we went back to look at those Hertfordshire adults and asked the question with DEXA again, where's their fat in elderly men and women, it was the same sort of distribution. Body fat was greater in the group that were low birth weight compared to high birth weight. The fat was more in the trunk than in the limbs, and it was associated with a reduction in lean body mass in those people. So it looks as if by changing diet, we can change individuals into more like the Indian thin fat baby around birth, and that this then plays out throughout their life. Now, Professor Simmons has introduced the concept of epigenetics to you, and so I want to show you briefly some of our reasonably recent epigenetic data. We have to see this, of course, against the background of the failure, I think I'd have to say, for the Human Genome Project and our studies, genome-wide screening, association studies, to find genes that account for a very high percentage of the risk of these diseases in the general population. Sure, there are some polymorphisms, some SNPs that are associated with risks with obesity later in life. The FTO gene is one. But if you look at this report in Science just a couple of weeks ago from the recent meeting of the American Society of Human Genetics, if you look at the red quote, the quest to find genes that strongly influence whether people will develop common diseases is turning out to be even more difficult than some researchers had expected. And so probably we need to be a little bit smarter and think more than just the fixed genetic inheritance that we all get, but the softer epigenetic inheritance that's passed on from generation to generation. How are we going to study that in our human populations? We can't take the pancreas and the liver and muscle out terribly easily. We decided to do that in tissues that have been archived from 2 of our prospective cohorts, in which both the placenta and the umbilical cord had been stored at birth. And then we went on to look at the phenotype of the children at 6 or 9 years of age. As Professor Simmons has told you, of course, you need to home in on some candidate pathways because the bioinformatics associated with epigenetic analysis is killing otherwise. And so one particular pathway we focused on is the PPAR, peroxisome proliferator activated receptor pathway, and retinoid receptor A. This is involved in responses at the genome level to many transcription factors and hormones and vitamins, vitamin D, corticosteroid, thyroid hormone, all have effects on downstream gene expression via this kind of process. And then when you think of it, the way that it responds to fatty acids, to prostaglandins, leukotrienes, one can see that this is a good candidate pathway to examine in our humans. And indeed, we have animal data that backs up that contention. And here, in summary, is the data that we published last year in diabetes that shows in 2 cohorts, one here, the PAH cohort, where the children were 9 years of age, These two over here, the Southampton Women's Survey, again another prospective cohort where the children were aged 6 years, showing the level of methylation at one of those CPGs that Professor Simmons has explained to you at birth, measured in the cord, against child's adiposity measured by DEXA either 6 or 9 years later. A dramatic effect. And in fact, when we first came up with this data, we realized how important it was to replicate the findings, which we did here, and replicated them again in that cohort here. So clearly, this is just the tip of the iceberg of an effect, but a very dramatic influence of this epigenetic mark at birth and adiposity in the child all those years later. But what's driving the epigenetic mark? Well, of course, we've got lots of information on diet in these women, and so when we examine in early pregnancy diets and then look at the epigenetic change at birth, we see that there's a pattern of dietary intake. It's close to an Atkins diet in these women. They have a low carbohydrate and a high fat and protein intake, although it's not high quality protein or good balanced fat. And that's associated with a higher level of methylation in the RXR methylation of the cords of their children at birth. And remember, it was that higher methylation that was associated with the increased adiposity of the child several years later. So a story is emerging of mother's dietary balance, and I should say that these are normal women in what is a very normal average city for Western Europe. Diet in those women affecting the tweaking of epigenetic marks in their children, which is affecting how they go on to develop in their early years. These studies are now being extended and repeated by several other groups, and there are 2 papers published this year which show 2 interesting developments. One is that it may be possible to look at such epigenetic marks in cord blood, which of course will change everything because this is very widely accessible. Actually, we know it's possible to measure it in the little Guthrie spots on the filter paper card. There's enough DNA to do that. The problem is that blood is a whole mixture of different kinds of white blood cells which respond very rapidly to immune challenges and other things in early life and throughout life. And so epigenetic changes in blood may not be representative of some of the other tissues. But it's a step in the right direction, and there's a lot more work to be done. The other thing that's appearing now is that some of the imprinted genes, here's IGF2, H19, which also shows some small epigenetic changes. So let's come back to these women, or a different lady here with her daughter, and ask the question about diabesity, if you like that term, the combination of obesity and diabetes being passed from generation to generation. Diabesity begets diabesity. We know that in the UK, many women of childbearing age, about 50% actually, are overweight or obese. And we know that that produces a whole range of epigenetic and metabolic effects on their offspring. Once again, I think we have to see this in the context of the normal population and ask, is there a point at which some combination of factors, mother's body habitus, her diet and so on, becomes pathological, or is this a graded phenomenon? And I think this data from Whitaker's group shows how that it clearly is a phenomenon that operates within the normal range. Here's the percentage of obesity in children at age 4 and maternal BMI. Obese women over here on the right, women that are probably too thin here over on the left, but graded across the whole range. So mother's obesity might be one driver. A second and very worrying one is the incidence of gestational diabetes. I don't know actually what it is in Dubai, but I know that in Singapore, Shanghai, some other parts of going further east from here, it's up to 1 in 5 pregnancies are complicated by gestational diabetes. And we know that that passes the risk on of diabetes to the next generation. But this data published last year, which you almost certainly can't read, is from Qatar, and it shows how various factors are linked to greater risk of gestational diabetes. It occurs more in Qatari nationals than in non-Qatari residents. It increases dramatically with age. it increases dramatically with housing conditions and to a certain extent with monthly income. So in this society, you might be forgiven for thinking that it's actually a disease of affluence and that this is all only an adult problem, until you begin to look at some of the other factors which clearly link it back to early development. A dramatic increase with parity with the number of children. Of course, with body mass index, as I've shown you, but then with other obstetric risks, abortion, stillbirth, and one would imagine preeclampsia, as well as diabetes too. So a very worrying trend. And we're beginning to see, once again, a very recent paper, that some of these processes from generation to generation in the gestational diabetes situation are associated with epigenetic changes. MEST is an imprinted gene that's involved in placental development and growth, and we're seeing changes there in infants that were exposed to gestational diabetes in pregnancy. So lastly then, what about interventions? What are we going to do about this? There are ways of rescuing the phenotype of the animal by giving pharmacological agents, endocrine agents, and even changing nutrition. But it's hard to see that in the newborn infant, these are necessarily things that are either easy or ethical or practical to do. So we need to think about more complex interventions, and they're going to involve wider social support. This may be a medical problem, but probably the solution is not a medical one. Are we going to find then a golden bullet which is going to somehow fix all this? I very, very much doubt it. And in fact, when we look from the Southampton Women's Survey and our other prospective studies at the cycle of risk from generation to generation, and I won't bore you with the data, but for every statement here we have statistically robust data to support it. We can see how it's the poor educational attainment of women and their poor diet and their lifestyle that leads to risk. They don't prepare for pregnancy, even if they plan it. When they are pregnant, they make minimal changes. They don't adhere to the infant guidelines, feeding scores such as they are. It plays out in phenotypic effects on their young offspring, less lean mass and more fat mass, becoming thin fat babies, if you like, even in Southampton. And then, of course, other aspects of neurocognitive development, poor IQ, certainly we see that at age 4, and so poorer educational attainment leading to the cycle being repeated. Where then might we intervene to prevent this horrible thing going on forever and ever? Well, one of the strongest drivers in our studies is women's educational attainment. And this data shows the percentage of a group of women who are consuming that poor diet, that imprudent diet that I showed you earlier on. And you'll see that for those who had a university degree, only 4% of that group consumed that diet. However, those who left school with no formal examinations whatsoever, it's about 55%. And add smoking to that as a risk factor, and it's 70%. So perhaps education may hold the key. And that really brings me to the conclusion that we feel that this place in between the young child and the woman, who will be one of the parents of tomorrow, if you like, adolescence may be the place that the Achilles heel, if you like, in the battle, where we may be able to produce an impact. You, as many of you I know, are neonatologists and pediatricians, and you might say, oh, well, that's it, then I'm not interested if he's going to talk about adolescence. But I would urge you to think about the continuity of care from one point in the life cycle to the next, and how actually what you can do and others can do here may prevent the problems that we're all seeing in pregnancy and the adverse outcomes later on. We know a little that such interventions can work. This is data, once again, published this year in the American Journal of Clinical Nutrition, that shows that the degree to which women adhere to a healthy diet, this is a Mediterranean diet, this is a diet designed to reduce the risk of hypertension, this is a low caloric type of diet, and the degree to which they adhere to it, as shown as quartiles here, is associated with a graded reduction in the incidence of gestational diabetes. So doing something pre-pregnancy can have an impact on that pregnancy and hence on the next generation. And for those of you who look at the Lancet, there was a nice series of papers earlier on this year focusing very much on adolescence. And it's an interesting thought that, I'll give you the headline first of all, that the proposed investments in adolescent health become investments not only in economic productivity and effective social functioning, but also in worldwide population health. Our young people and all of us will benefit. That was their banner headline. And it's an interesting thought that in the world, more than 25% of the world's population are now aged between 10 and 24. And if you look at the data, you can see that actually their health has improved less than younger children over the last 50 years. We've rather been letting this group of the population down. It's a sensitive period in development when, of course, with puberty and brain maturation, we can change behavior, and that can, of course, have major impact on health and well-being. Even more, those of you who have adolescent children know that they're very powerful agents for change in the family. What they want to do very often carries the day. And so we have some possible advocates, people who can fight with us in this battle if we can bring them on side. Of course, the strongest determinants of adolescent health are wealth, no surprise there, inequality, but also access to education. That was one of the conclusions from these 4 papers in the Lancet. So I must stop. What I hope I've shown you is that the DOHAD idea, Developmental Origins of Health and Disease paradigm, provides a new model for non-communicable diseases, things that develop gradually, where the risk racks up as time goes on. And there's therefore a very strong case for interventions in early life to prevent these diseases. How do we do it and in whom? Well, we're beginning to develop biomarkers of NCD risk. Some of them are epigenetic, some of them are other, anthropometric, and so on. We can measure them, and so we can target interventions. We can do studies to measure their efficacy and their cost effectiveness. We've moved, I think, from focusing just on low birth weight, which is a problem, but to be able to see this problem as something that affects NCD risk in a graded way across the entire range of development. Everybody in the world is as a risk of NCDs. All that matters to you and me, and actually to the population group as a whole, is where are we on that trajectory risk? How high is it, and how fast is it increasing? What we're saying is not that these diseases start in development, but that actually development influences our responses to later challenges. And that's sometimes a concept that's difficult to get across to our colleagues. And then I believe that preconception may be a critical period in terms of influencing the health of parents-to-be, which means a focus on adolescent girls and boys too, because of course it's a partnership which we need. I think is that we can begin to think about interventions perhaps in adolescence that can have an impact on some of these biomarkers that we can measure at birth in a period of perhaps 3 to 5 years. So rather than focusing, as David Barker and other colleagues working at the end of the last century did, on intrauterine life having an impact 50 to 60 years later, we can begin to see how we might make the health of children born tomorrow and next year and the year after healthier. And then lastly, for those of you that are interested in DOHAD, I'd urge you to come along to our next World Congress, which is in Singapore. Some beautiful architecture there. I'm not sure that it's as nice as Dubai, to be honest, but there we are. Some very good science, and a meeting that's going to be across the board from basic science to epidemiology and public health, and very much focusing on the issue of taking science to policy. So I hope that in following on Professor Simmons's talk on mechanisms, I've given you some insight into some of the implications of this concept. Thanks very much for listening.